Ever wondered how drugs work at the cellular level? How do they interact with our body's receptors? These are questions that puzzle many. We're about to embark on an exploration, unraveling the mysteries of receptor kinetics. This fascinating aspect of pharmacology reveals how drugs and our bodies interact on a microscopic scale. It's a journey through binding, activation, desensitization, resensitization, internalization, and recycling. Strap in, it's going to be a wild ride. The answers lie in understanding receptor kinetics. Receptor kinetics is the study of the rates at which reactions occur in biological systems. Now, let's dive into the heart of the matter. Receptor kinetics, in its simplest form, is all about timing. It's the study of how quickly or slowly reactions happen within biological systems. This may sound a bit technical, but think about it this way. Imagine you're cooking a meal. The time it takes for your ingredients to cook and the order in which you prepare them can greatly affect the final dish. The same goes for reactions within our bodies. In the realm of pharmacology, receptor kinetics plays a crucial role. Drugs are essentially ingredients that we introduce into our bodies. The speed and sequence of their interactions with our biological systems can determine their effectiveness, side effects, and overall impact on our health. Receptors are like the chefs in our biological kitchen. They are proteins typically found on the surface of cells, and they interact with substances, like drugs, to trigger specific responses within the cell. The dance between a drug and its receptor is a carefully choreographed ballet, with each movement and pause playing a vital part in the overall performance. Understanding receptor kinetics helps us predict how a drug will behave in the body. Will it bind quickly to its receptor, triggering an immediate response? Or will it bind slowly, leading to a gradual effect? These are the types of questions that receptor kinetics can help answer. Furthermore, receptor kinetics can shed light on the drug-receptor relationship over time. For instance, will the drug remain bound to its receptor for a long time, sustaining its effect? Or will it detach quickly, causing a short-lived response? These insights can greatly influence the design of new drugs and the optimization of existing ones. So receptor kinetics is not just about the when and how fast. It's also about the how long and the what next. It's about piecing together a dynamic picture of drug-receptor interactions, and ultimately, it's about understanding how we can manipulate these interactions to improve health outcomes. So, receptor kinetics is a fundamental concept in understanding how drugs interact with our bodies. The first step in receptor kinetics is binding. Let's dive into what binding actually means. In the context of pharmacology, binding refers to the interaction between a drug, which we often call the ligand, and a receptor. This interaction is a lot like a lock and key mechanism. Imagine the receptor as a lock, and the ligand as the key. When the key fits perfectly into the lock, the door opens, initiating a series of events that we'll delve into in the later scenes. Now, not all keys are made the same, right? Some keys may fit into the lock but won't turn. Others might slide in but get stuck halfway, while some won't fit in at all. It's the same with ligands and receptors. Some ligands fit perfectly into the receptor, triggering a full response. We call these agonists. Others may bind to the receptor but only trigger a partial response. These are partial agonists. Then there are those that bind to the receptor but don't trigger any response at all, blocking other ligands from binding. We refer to these as antagonists. So why is binding so important? Well, the binding step determines the efficacy of a drug. Efficacy is the ability of a drug to produce a therapeutic effect. If a drug binds perfectly to its receptor and triggers a full response, it has high efficacy. On the other hand, if a drug binds to the receptor but doesn't trigger a response, it has zero efficacy. Therefore, understanding the binding process is crucial in drug development and therapeutic treatments. In a nutshell, binding is the first and perhaps the most critical step in receptor kinetics. It sets the stage for all the subsequent steps, dictating how a drug will interact with the body's receptors and the kind of response it will elicit. After binding comes activation, and what a fascinating process it is. The second step in receptor kinetics, activation, is a critical player in the pharmacodynamic theater. Imagine the receptor as a lock and the drug as a key. When the key slips into the lock, that's binding. But the story doesn't end there. The lock needs to be turned, and that's where activation comes into play. What does this turn of the key look like in the world of molecular pharmacology? It's all about conformational changes. Essentially, when a drug binds to a receptor, it prompts a change in the receptor's shape. This change is what we refer to as activation. Once the receptor is activated, it sets off a series of events, much like dominoes falling in a line. This cascade of events is known as signal transduction. The activated receptor initiates a chain reaction, transmitting the signal from the drug to the cell's interior. This transmission can either stimulate or inhibit specific cellular functions, depending on the nature of the drug and the receptor. Now here's where things get a bit interesting. The relationship between activation and drug effect isn't always straightforward. In some cases, a drug may bind to a receptor and activate it, but not produce a full effect. These are known as partial agonists. On the other hand, some drugs can bind to a receptor and prevent its activation, even in the presence of other activating substances. These are called antagonists. But for our standard model, let's stick to full agonists. These are drugs that bind to a receptor, activate it, and produce a maximal response. The activation of the receptor by a full agonist is what ultimately triggers the drug's effect. It's like turning the key in the lock and opening the door to a whole new realm of pharmacological possibilities. So to sum it up, activation is the turning of the key. It's the process that follows binding, involves conformational changes in the receptor, triggers signal transduction, and ultimately leads to the drug's effect. Activation is what turns the key in the lock, triggering the drug's effect. The next steps in receptor kinetics are desensitization and resensitization. Let's dive into these fascinating concepts. Desensitization is a process that occurs when a receptor is continually exposed to an agonist. In response to this sustained exposure, the receptor becomes less responsive to the stimulus. It's like when you first move to a busy city. Initially, the noise is overwhelming, but over time, you become less sensitive to it. You've essentially desensitized to the constant buzz. This process is crucial in the context of drug administration. With sustained exposure to a drug, the body's receptors may become desensitized, leading to a decrease in the drug's effectiveness. This is a primary factor in the development of drug tolerance, where higher doses of a drug are needed to achieve the same effect. Now, let's talk about resensitization. This is the flip side of the coin. It's the process by which the receptors regain their sensitivity after becoming desensitized. It's like when you leave the city for a while and then come back. The noise seems loud again because you've resensitized to it. Resensitization is particularly important in the context of drug dependence. When a drug is removed, the body's receptors can become hypersensitive, leading to withdrawal symptoms. Within these processes of desensitization and resensitization, we also have the concepts of receptor downregulation and upregulation. Downregulation is a decrease in the number of receptors, often a response to prolonged exposure to an agonist. Upregulation, on the other hand, is an increase in the number of receptors, usually in response to a lack of agonist. These processes are another layer of complexity to how our bodies respond to drugs. They can play a significant role in drug tolerance and dependence, as well as the effectiveness of drug therapies. Desensitization and resensitization are the body's way of maintaining balance in response to drug exposure. The final step in receptor kinetics is internalization and recycling. Imagine your cells as a bustling city and receptors as the city's workers. After a long day of hard work, they need a break. That's where internalization comes in. It's the process by which receptors, after being activated and possibly desensitized, are brought back into the cell. It's like our city workers going home for a good night's rest. But why do receptors need to be internalized? Well, it's a clever mechanism to regulate receptor sensitivity. By taking activated receptors off the surface, the cell can control how many receptors are available for binding. It's a bit like the city turning off the lights after a certain hour to conserve energy. Now, what happens to these internalized receptors? Do they just stay at home, never to return to work? Not at all. Many of them undergo a process called recycling. In recycling, the receptors are transported back to the cell surface, ready to be activated again. It's as if our city workers, after a night of rest, are heading back to work. Interestingly, not all receptors are destined for recycling. Some receptors, once internalized, are sent to a part of the cell called the lysosome. There they get degraded or broken down. It's as though some of our city workers, after a long career, retire and leave the workforce. So you see, internalization and recycling are crucial in maintaining the balance of receptor activity. They ensure that the cell doesn't become too sensitive or too insensitive to a particular signal. It's a bit like how a city needs to balance the number of workers it has. Too many, and resources get stretched thin. Too few, and work doesn't get done. And what about drugs? How do they fit into this picture? Well, long-term drug use can alter this balance. It can lead to more receptors being internalized and fewer being recycled, making the cell less responsive to the drug. It's akin to the city workers going on strike after being overworked. Internalization and recycling help the body to adapt to long-term drug use. Let's recap what we've learned about receptor kinetics. We kicked off with the intriguing concept of receptor kinetics, a fascinating dance of molecules that governs how drugs interact with our bodies. We've journeyed through the intricate steps that make up this process, and it's time to consolidate what we've learned. First off, we talked about binding, the initial step where drug molecules latch onto specific receptors. This is akin to a lock and key mechanism where the drug, the key, fits perfectly into the receptor, the lock. This specific interaction ensures that each drug affects only its intended target cells. Following binding, we move on to activation. This is the moment the magic happens. Once bound, the drug activates the receptor, triggering a cascade of biochemical reactions within the cell. It's like flipping a switch that sets off a series of events, leading to the therapeutic effect we desire. But what happens when the cell is constantly exposed to a drug? Enter desensitization and resensitization. Desensitization is the cell's way of protecting itself from overstimulation. It reduces its responsiveness to the drug over time. But don't worry, this isn't a permanent state. The cell can resensitize itself, restoring its ability to respond to the drug. It's a bit like taking a break from a loud noise, then gradually getting used to it again. Lastly, we explored the steps of internalization and recycling. After a while, the cell might decide to pull the receptor inside, away from the drug. This process, known as internalization, can help the cell control its response. But this isn't the end of the road for the receptor. It can be recycled back to the cell surface, ready to interact with the drug once more. It's a clever way the cell maintains balance, like a DJ adjusting the volume on a sound mixer. And there you have it. From binding to recycling, we've navigated through the dynamic process of receptor kinetics, understanding how drugs interact with our bodies to bring about their effects. So, that's receptor kinetics in a nutshell. Now that you understand receptor kinetics, you might be asking, why does it matter? Well, let's dive into it. Receptor kinetics is like the secret language of our bodies, a code that helps us understand how drugs interact with our system. This knowledge is crucial in the field of pharmacology, especially when it comes to drug development. Imagine you're a pharmacologist, working tirelessly to develop a new drug. You've got the perfect molecular structure, but how do you ensure that it will effectively bind to the right receptors? Or, how do you predict the body's response to the drug? The answer lies in understanding receptor kinetics. Receptor kinetics provides a detailed roadmap of the drug-receptor interaction. It allows us to predict how a drug will behave once it enters the body. Will it bind quickly or slowly to the receptor? Will it activate the receptor instantly or take its sweet time? Will the receptor become desensitized or remain active? All these questions can be answered through receptor kinetics. Moreover, receptor kinetics also helps us understand drug interactions. For instance, if two drugs bind to the same receptor, they might compete for binding sites. This could potentially alter the effectiveness of these drugs. By understanding the kinetic properties of these drugs, pharmacologists can predict these interactions and adjust the dosages accordingly. But the influence of receptor kinetics extends beyond just drug interactions. It also plays a vital role in predicting drug responses. The speed at which a drug binds to a receptor, or how quickly the receptor becomes activated or desensitized, can significantly impact how a person responds to a drug. This can be the difference between a drug providing relief or causing adverse effects. In essence, receptor kinetics is a key player in ensuring the safety and effectiveness of drugs. It provides the necessary insights to fine-tune drug design, anticipate drug interactions, and predict drug responses. Without it, we'd be navigating the complex world of pharmacology blindly. So the next time you pop a pill or get a shot, remember the intricate dance of receptor kinetics that's happening within your body. It's this dance that ensures the drug does its job, helping you feel better. In conclusion, understanding receptor kinetics isn't just a fascinating intellectual exercise. It's a vital tool in the creation of effective and safe drugs. As we continue to advance in the field of pharmacology, the importance of receptor kinetics will only become more apparent. Understanding receptor kinetics is essential for the development of effective and safe drugs.